In this chapter, we're going to talk about the regulation of gene expression. And there's a few different topics we'll address, but we're going to start with how the little guys do it first, how bacteria or prokaryotes regulate their genes. Of course, depending on which book you're using, it's in chapter 18, and those are the pages that you can utilize for this. So let's first talk about bacterial metabolism. Think about how bacteria are just single-celled creatures, and they tend to live inside of us or around us or on us. So they're going to get their nutrients from the environment in which they're found. Sometimes those nutrients are plentiful, and sometimes they're not. Sometimes, let's say, they need to make certain proteins. Those proteins in their environment or amino acids that they need for those proteins might be in abundance. So they don't actually have to make the proteins themselves. They can just digest them and get them. Or sometimes there aren't those nutrients available. So sometimes bacteria might need to actually make, say, like an amino acid if it's not in its environment where it can just feed off of it. So bacteria have to respond very quickly to changes in their environment. Otherwise, they're not going to get what they need, or the other opposite end of the spectrum is that they might actually be wasting energy making something that's already there. So this goes back to a very long time ago when we talked about regulating metabolism. Remember feedback inhibition? This is a good time to review this concept. In this case, this was one of those body mechanisms that keeps us from making too much of a good thing. So tryptophan is an amino acid, and when we have enough of it, why should we worry about making more? So as it goes here, tryptophan is actually an inhibitor to the first enzyme that would create it. So when you have enough tryptophan in your system, it can actually act as that allosteric inhibitor, meaning it binds to a spot on the enzyme number 1 here, opposite of the active site, which then shuts down this enzyme so that we stop making the enzymes to create tryptophan. So feedback inhibition, remember it's a metabolic pathway, and again it's a way to stop the making of certain enzymes in a pathway so that we're not wasting energy. And this works well for us, but if you think about it, we have these enzymes that are in our cells. They're just being blocked right now. So that means, if you go back to the beginning on how we make enzymes, that we actually went to all that energy to transcribe the DNA and then send that mRNA to the ribosome, and then go to all the work to putting those amino acids together to actually assemble all of these enzyme proteins here. So then those enzymes are just floating around in the cell right now, not being used because they're being blocked by the presence of tryptophan, the amino acid. Well, so that's kind of a waste of energy in that sense, if you think about it, that you went to all the work to create those enzymes just to kind of let them float around and, well, not do anything. So think about it also in this way. Is there a way to actually block the transcription of those genes so that we don't go to all that work to making those enzymes in the first place if we're just going to block them anyway and let them sit around not doing anything? Well, of course there is. Bacteria have ways of regulating their genes called gene regulation, where they can actually block the transcription of the gene in the first place, thereby saving energy, at least here in the tryptophan pathway, and not making those enzymes in the first place unless they really need to. So gene regulation in bacteria, very important. They don't want to waste energy, and they want to make use of the nutrients in their environment when they can. So they can regulate gene transcription by turning genes on and turning genes off. So we're going to look at two different examples. One of them is for the tryptophan pathway, where this will be an example of turning genes off, and then one for the lactose pathway, where we turn genes on. In the tryptophan pathway, if a bacteria has enough tryptophan, then it really doesn't need to make those enzymes in order to build more tryptophan. That would be a waste of energy. And in the lactose pathway, if a bacteria encounters new sugar, hey, that's an energy source that we want to utilize. And, well, lactose is an actual sugar. So it would need to start making enzymes used to digest it. But then again, you also have to think about it that it doesn't want to make those enzymes unless lactose is actually available to do so. So when you put groups of genes together in order to regulate the genes, it's called an operon. And bacteria can make use of operons with the two examples I said we were going to look at, the tryptophan and the lactose operons. So by definition, an operon is basically just a bunch of genes grouped together, and they have related functions. So if you group them together, it makes it easier to regulate them. If you want to block them, and they're all in kind of this one little spot along the genome, it's easier to control that. Or if you want to activate them, and again, they're kind of in one spot on the genome, it makes it easier to regulate them and activate them when needed. So the different components of an operon first includes what we'll call a promoter. The promoter is the RNA polymerase binding site. So remember what a polymerase does? Basically, it builds nucleic acids. So we talked before about DNA polymerase, how there's a few different kinds of DNA polymerase, and it builds DNA by adding nucleotides. Well, RNA polymerase does basically the same thing. It needs to bind to a site and then start adding RNA nucleotides to build, such as an mRNA transcript. And if we group all of our genes for a metabolic pathway in basically the same area, then we only need one promoter to control the transcription of all those genes in that operon. So they can all be transcribed as one single mRNA unit. The operator is the actual DNA binding site of a repressor protein. A repressor can block transcription so that you don't make those proteins. So if we look at our diagram below, it is the diagram for the tryptophan operon, often nicknamed the TRP or trip operon. So this whole area here belongs to the tripopteron. It's just a bunch of genes grouped together with related functions. So in order to make tryptophan, we would need to transcribe all 5 of these genes, TRPE, D, C, B, and A. We would need all those proteins, which are basically enzymes, to make tryptophan. But hey, if we already have enough tryptophan in our systems, then why make those enzymes? So that's why we need this operator. So we group them all together so we can control them. And as you can see, if we do go ahead and we make an mRNA transcript of the genes, that mRNA transcript has a complementary copy made from DNA of all these genes right here. All 5 of them are in this mRNA transcript, and remember that would then travel to the ribosome to be made into proteins. All right, we have the actual promoter region here. Again, that's our polymerase binding site and can control the transcription of all these genes in this area. And this symbol here shows our RNA polymerase, who would need to move along this whole area to make an mRNA transcript. And then we also have the operator in yellow, and remember, structure determines function, so this operator has a certain shape, and you'll see why in a little bit. All right, let's look at the repressor protein a little more closely. As you can see here, again, this yellow object here with the notches, that was our operator. And the red thing is our repressor protein. As we can see, the repressor protein is shaped just right in order to bind into the operator. And if that repressor protein is bound into the operator, then, well, here's our RNA polymerase. Our RNA polymerase cannot move along the DNA in order to make an RNA transcript. So it's basically blocking transcription if it's in place. So our protein looks like this. If we go further and take a look down here, the repressor protein is in red, and it has a certain shape, and when it's in this shape, it does not fit into the operator, so it cannot block transcription. But when tryptophan is present, tryptophan, this little orange hexagon and pentagon, when tryptophan is present, it binds into the repressor protein, thereby changing its shape. Remember, that's a conformation change. And then it does fit into the operator and blocks transcription, which makes sense because if we have tryptophan present, we don't want to have to spend energy making those enzymes. So the tryptophan will bind into the repressor, then the repressor will bind into the operator, and then block RNA transcription by blocking the RNA polymerase. So let's take a look at how all this works. All right, now I've started up my PowerPoint here to play along some animations for you. So here's our tryptophan or trip operator, or operon, I should say. And here's some of the genes that are in the operator in DNA. Notice they're all grouped together in the bacterial genome. And then up front here's our RNA polymerase where it's binding to the promoter. And we discussed before how the promoter is usually a TATA box, the TA. There's the operator right there. And if I click, there it goes. There's nothing blocking our operator, so our RNA polymerase is actually able to transcribe the gene, which then makes an mRNA transcript, which then goes to the ribosome, which will then be turned into proteins or the enzymes needed for this metabolic pathway. In this case, we're talking about the manufacture of tryptophan, but we need the enzymes in order to do that. So if we don't have tryptophan in our environment, we're going to have to make it. So this is how we do that. All right, if I do another click though, let's say that we have tryptophan in our environment. So now we don't need to transcribe those genes anymore. That would be a waste of energy. So now the repressor protein has bound to the operator. Oops, and as you see here, it blocks the RNA polymerase, which is what we wanted in the first place. So essentially, we have turned this gene off, or this operon off. If we turn that operon off, then we don't get an mRNA transcript, and we don't make those proteins. This is an operon found in E. coli bacteria. Remember how prokaryote genomes look, that they are not in a nucleus because prokaryotes do not have nuclei. The genome is basically a circular chromosome floating around in the cytoplasm. And so now if you can visualize this, that we're actually grouping certain genes together. In this case, all the genes for the metabolic pathway to make tryptophan are grouped together with the promoter and operator out in front so that we can regulate these and only make the enzymes to make tryptophan when we need to, when it's lacking from our environment. Take a look at this again. There goes our RNA polymerase. Again, we're making mRNA transcripts. Those will go to the ribosome and will assemble those amino acids into the proper enzymes in order to make tryptophan. Oh, plenty of tryptophan being made. TRP means tryptophan. All right, so we've got plenty of tryptophan in the environment. So now we have bound to the repressor protein, and we have then bound to the operator. This causes a conformation change and blocks our RNA polymerase. All right, so no RNA polymerase means no mRNA transcript, which means no enzymes to make tryptophan anymore. Therefore, the bacteria will start using their tryptophan and probably use it up. All right, so what happens when tryptophan is present? Take a look. Well, we don't need the enzymes if that's the case. Remember, tryptophan is an allosteric regulator of the repressor protein. So here's our tryptophan, and it binds into a site opposite of the active site, which is up here, which causes, as you can see from here to here, a conformational change in that repressor protein. All right, here's another one. Let's take a look at the opposite type of operon called a lactose operon. This one's called an inducible operon. Again, this is a digestive pathway model where our bacteria are going to react to what's in their environment. So let's say that they come across an environment that has plenty of lactose. Maybe these are bacteria that live inside your gut and you just drank a glass of milk. Well, there's lactose in that milk and they could make use of that lactose as an energy source. But lactose is a disaccharide, so it does need to be broken down in order to utilize the two monosaccharides that make up lactose. So to do that, that would be great because that's a great energy source. But again, you don't want to make enzymes to break lactose down if lactose is not in the environment, because that would be a waste of energy. So as you see with this is called the lac operon or lactose operon, that it looks pretty similar to the last one we just saw, that everything is kind of right here in one area in the bacterial genome. We have our promoter, which is the site where RNA polymerase binds, and it's in the form of a TATA box, the TATAs. We have a repressor or an operator here where the repressor protein can bind. It looks like at the moment the repressor is in place. Then we have the genes that are involved in the breakdown of lactose. So in our last one, in the tripopteron, we were actually making enzymes to build tryptophan. In this case, we have genes that are going to make enzymes to break down lactose. All right, so the repressor's in place. It looks like it just blocked RNA polymerase. Oh, but wait a minute. Maybe you just drank a glass of milk and there's plenty of lactose present. There they are in the form of the hexagon, even though lactose is actually a disaccharide. But we'll play along here that it's just a hexagon for now. All right, so when lactose is present, it binds to the repressor. And look at what it does. It causes a conformational change in that repressor protein, and oops, there it goes away. And there's our RNA polymerase. Now it's able to bind, and there it goes, transcribing those genes. I just like watching that. All right, so if we transcribe the genes, that means we make an mRNA transcript, which then can go to our ribosome and be made into proteins or enzymes that will help the breakdown of lactose. And here's another diagram to show that. Remember, bacteria need to make lactose digesting enzymes. So then we'll turn on this lactose operon and we'll actually make our mRNA transcript. So all the parts are shown here. Again, there's our RNA polymerase bound over the promoter. There's our operator. Here's our repressor protein in red. And in this case, the green is the lactose, which can bind to the repressor protein and get it to let go of the operator so that RNA polymerase can get through. And then we can make our enzymes. Lactose is also an allosteric regulator or inhibitor of the repressor protein. It binds to a site opposite of the active site. We'll give a little shout out to these guys for discovering the lac or lactose operon. And they actually coined that phrase, operon, which is the group of genes that regulate a certain pathway. All right, so the repressible operon, this is usually for anabolic pathways. Remember, anabolic pathways are those that build, just like anabolic steroids can build. Anabolic pathways are used to build molecules like the building of tryptophan. An inducible operon, like the one for the lac operon or lactose, functions for catabolic pathways. Catabolic means breaking down. Just like my cat breaks down my Christmas tree every year, a catabolic pathway can build enzymes to break something down, like the digestion of lactose. And remember, we only want to utilize these molecules when we need them so that we can save energy. So the operons allow us to turn things on or shut them down when we need to, especially if we group them all kind of in one area of the bacterial genome. Here's some videos that can really show this, plus some awesome animations, like Mr. Anderson with his Bozeman Science videos. He's from Bozeman, Montana, so he calls it Bozeman Science. Those are all on YouTube. I would definitely take a look at those. And then, of course, the Amoeba Sisters, who really know how to take things and break it down into smaller components to help get the information through. And don't be repressed. Be induced to ask questions. We didn't cover everything in the Chapter 18 learning targets yet, but be sure to take a look at the objectives in the chapter. Our actual AP or college learning targets kind of take all those littler ones and put them into bigger categories. If you like, I can get you a copy of this, but here's those learning objectives right here. There's 4 of them, and here they are, 1, 2, 3, 4. And if you want to break them down even further into the essential knowledge, you can read those right here and see which ones were covered in this lecture.